It was the most excruciating pain I've ever felt. Someone had hit me on the side of my head. My eyes couldn't focus on where they were. I started vomiting, which only made it worse. I don't know how long later I took some painkillers, but it made no difference. All I could do was sit in the dark and wait for this violent attack to end. How did this violent attacker get into my home? If only there was some way of stopping them from getting in. This picture was created by a person living with migraine, and it matches my attack closely. Did you know more than 1 billion people worldwide are living with migraine? And it is the second leading cause of years lived with disability. Yet, we still don't know how a migraine attack occurs. What we do know is, if migraine patients are infused with a particular small protein, a migraine-like attack is triggered. This protein is called calcitonin gene-related peptide, or CGRP. Now, multiple drugs have been developed to target CGRP and its receptor. These CGRP-specific drugs have been life-changing for migraine patients. But they don't work for all patients, and we still don't know the long-term effects of these drugs. To answer these questions, it's important to understand how these drugs are acting at the cellular level. This is where my research comes in. Some of these drugs have been designed to bind and stop the activation of the CGRP receptor, which acts as a messenger, relaying information into the cell. But there's another receptor, the amylin-1 receptor, which can also relay information from CGRP into the cell. My work engineers cells to create these two receptors so I can compare how they behave in the presence of CGRP pathway blocking drugs. My results have shown that even though these drugs were designed to target the typical CGRP receptor, they can also act on the amylin-1 receptor. So what does this mean? Is the ability to also act on the amylin-1 receptor helpful or harmful? More research is needed to fully understand what is happening during a migraine attack, and there's still a lot we don't know. But my work provides insight into how these drugs are acting at the cellular level, which then can be applied to creating better therapeutics. My work is a small piece in a larger puzzle that will hopefully end with fewer disabling migraine attacks. If we can stop these messengers, maybe we can stop these home invasions.